Cardiac contractile cells regulate the life-sustaining beating of the heart. Today we will talk about the action potentials of these cells and how the ion movements involved in this process allow the heart to contract and relax in a synchronous fashion. Cardiac contractile cells are activated by the pacemaker cells of the heart. The action potential in the cardiac contractile cells undergoes 5 distinct phases. These phases include the rising phase, or phase 0, the peak phase, or phase 1, the plateau phase, or phase 2, the falling phase, or phase 3, and the resting phase, or phase 4. We will now discuss each phase in more detail. Phase 4, also known as the resting phase, constitutes the beginning and the end of the action potential of cardiac contractile cells. As shown in this illustration, both sodium and calcium channels are closed in this phase. Potassium channels are open in order to maintain the resting transmembrane potential of -90 millivolts by the constant leaking of potassium from inside to outside the cells. These potassium channels are known as leaky channels. As the name suggests, during this resting phase, the heart is not contracted. The next step in the action potential process of cardiac contractile cells is phase 0, or the rising phase. During the rising phase, electrical activation from the pacemaker causes the opening of voltage-gated sodium channels. This depolarization occurs when sodium rushes into the cell rapidly, causing an increase in membrane potential. As the threshold rises to the potential of -70 millivolts, the inward sodium movement becomes self-sustaining. In fact, the potential increases from the resting -90 millivolts to about 20 to 30 millivolts with the sodium influx. As depicted in this illustration, phase 0 is the gray strip climbing vertically to represent the drastic change in membrane potential. During this process, long-type calcium channels also open when the potential reaches -40 millivolts, causing calcium influx into the cell. The membrane potential movement in the positive direction is directly correlated to the influx of these positive ions. The next phase of cardiac contractile cells is the peak phase, or phase 1. In this phase, potassium channels open. These transient channels support potassium efflux out of the cell. This potassium loss causes the transmembrane potential to decrease back to 0 millivolts from the 20 to 30 millivolt increase of phase 0. This is known as a slight repolarization of the potential. This is depicted in the illustration by the downward movement of the action potential before phase 2 begins. The next phase is known as phase 2, or the plateau phase. In this phase, the long-type calcium channels from the rising phase are still open, allowing the slow influx of calcium. The leaky potassium channels are also open, which allows the efflux of potassium. The concomitant influx and efflux of both positively charged ions allows for the transmembrane potential to be maintained. This is known as the plateau phase. This plateau lasts approximately 175 milliseconds. This is vastly different than the action potentials of both neurons and skeletal muscle that last approximately 1 to 2 milliseconds. The length of this plateau phase is an important factor for the specialized function of cardiac contractile cells. In fact, it is the plateau phase length that allows the proper communication between the action potential of the cardiac contractile cells with the actual contraction of the cardiomyocytes upon which they exert their effect. As you can see in this illustration, by the line marked 2, the plateau phase is appropriately named as it forms a physical plateau of membrane potential on the graph provided. The next phase is the falling phase. During the falling phase, or phase 3, calcium channels close gradually while leaky potassium channels remain open. This is depicted in the illustration by the falling red line marked 3. This means the efflux of positive potassium ions outweighs the halting influx of positive calcium ions. This repolarization returns the transmembrane potential to the resting -90 millivolts to prepare for the next action potential. This, of course, is the return to the resting phase 4 mentioned first in this presentation. To explore the careful regulation of cardiac contractility and the effect that alterations can have, we will now discuss a study completed in 2019. As discussed, the ion movement is vital for proper cardiac function. So, chemicals and disease states affecting these ions can pose problems for normal contractile activity. The study we will discuss today is titled Cardiac, Electrical, and Contractile Disorders Promoted by Anabolic Steroid Overdose are Associated with Late Autonomic Imbalance and Impaired Calcium Handling. In exploring this study, we will learn the effect that anabolic steroid overdose has on heart function in relation to calcium ion sensitivity and action potential. In the control group of this study, male Wistar rats were administered the placebo vehicle CTL. On the other hand, the experimental group of male Wistar rats were administered an anabolic steroid known as nandrolene decannoate or DECA, DECA. The parameters of the study were measured at 2, 4, and 8 weeks in both groups. The hypothesis of this study was that anabolic steroid overdose in the form of DECA would cause both mechanical and electrical dysfunction of the heart. For the results of this study, first we will look at Figure A. In Figure A, the white bars represent the CTL or control group, and the black bars represent the DECA or experimental group of rats. The vertical y-axis is the calcium loading compared to the time on the horizontal x-axis. In the experimental group, the rats that were administered the anabolic steroid DECA had decreased calcium loading at each measured time of 1, 3, and 5 minutes, compared to the control group. Furthermore, Figure B depicts the same bar representations of each group with calcium release on the vertical y-axis and amount of caffeine administered on the horizontal x-axis. When administered the same amount of stimulating caffeine, the experimental group released much less calcium in response than the control group. Finally, Figure C illustrates the much increased isometric tension of cardiac fibers in the experimental DECA-exposed rats compared to the tension of control group rats at different concentrations of calcium levels. As discussed previously, calcium plays an important role in the action potential of contractile cells. As in the previous figures, the DECA experimental group is still expressed by the black bars, and the control CTL group by the white bars. In Figure A, we see that heart rate was not significantly different in either group at weeks 2, 4, or 8. Furthermore, resting membrane potential as well as action potential amplitude were also not significantly different between the control and experimental groups. However, Figure B does exhibit a significant difference with an increased QTC interval for the experimental group at 2, 4, and 8 weeks. Furthermore, major differences in action potential duration were also observed. Figure C represents the action potential duration at 30% of repolarization. Figure D represents the action potential duration at 90% of repolarization, and Figure E represents the triangulation between the two. Ultimately, anabolic steroid abuse causes both mechanical and electrical impairment of the heart. Prolongation of both the QTC interval as well as the action potential duration can have detrimental effects on the heart's ability to function. Alone, either of these problems can cause arrhythmias and tachycardia. In combination, this risk is even greater. Arrhythmias and tachycardia can cause the heart to work much harder and lead to mild symptoms like racing heart and dizziness, or even more problematic and deadly consequences like stroke, heart attack, and even death. Furthermore, the effects on calcium ion balance can affect action potentials as well, since this is a major ion in the proper function of the action potential phases of contractile cells, as well as many other cells throughout the body. Finally, increased sensitivity to calcium ions can also cause arrhythmias. This study is just one example of the major effect that externally introduced factors can have on proper heart function. In conclusion, cardiac contractile cell action potentials and their unique phases are extremely important for proper cardiac function. The precise opening and closing of ion channels in response to stimulation, as well as the influx and efflux of the involved ions to influence membrane potential, are crucial components of this system. Furthermore, The unique plateau phase of these contractile cells ensures proper cardiac excitation, contraction, communication to the cardiomyocytes. This communication is crucial for rhythmic, controlled, and steady heartbeat function, and therefore imperative for human life.